So just before starting, just let you know that this is the space here, or maybe here in the corner, that you can put your video, just relax and see the presentation without the need of moving the video to see video and stuff. So we know that webs are really interesting thing in spiders, certainly like the most striking feature of spiders. But we need to remember that most of species do not build webs at all, like the jumping spiders, the wandering spiders. Here we have I think the most recent phylogeny of spiders with the ancestral state reconstruction of web. I'm not gonna go into details here, but certainly you can see more in the next talk, I think. But what I would like you to pay attention here is in this clade, the RTA clade, which comprises 50% of all spider diversity. And inside this clade, we have another clade, the Dionycha, that are 60% of RTA and 30% of all spider diversity, and they do not build webs at all. So they have this amazing diversity inside Dionycha, and a lot of ecological. They are ecological diverse. They are taxonomically diverse, and there's like a lot of character systems just waiting to be studied and to understand their evolution. For example, the tapetum of Gnaphosides, they're oriented in this 45 degrees, and this probably helps to have a better perception of the polarized light in the sky and to orient themselves. In other spiders, this tapetum, the tapeta are parallel to each other. We also have other features, like although the Dionycha do not build webs, they have a very impressive variation in the spinning organs. And one impressive case is the Gnaphosides. They have this enlarged Piriform Gland Spigot that they use to attach the prey to the ground. And of course, I should not forget to mention the absence of the third claw. So the plasomorphic state in spiders, three claws in the tarsus, but Dionycha have lost the third claw. And also there's associated with this loss, or associated to the food of spiders, have a lot of interesting characters, like the claw-tuft and the claw-tuft base. So sometimes you have this base that are movable, and another times the bases are fixed, but you have this clasping mechanisms that help to grasp the seta and move the seta. So all those things help the spiders to stick to surface, to smooth surface. So this was thought to be a very important characters of Dionycha, very important for Dionycha evolution. It's thought to be the great silk alternative. So they use to adapt to this new new environments, and also to prey, to help to catch the prey. But all this, to understand all the evolution of these characters of Dionycha, we need to understand better the phylogeny. So we lack a robust phylogeny of knowledge about Dionycha phylogeny. Dionycha rarely include in phylogenetic studies, even the most recent phylogenomics. The group are represented only by few specimens, few families. So we have some interesting things, those some families that were always thought to be Dionycha based on morphology. So all those families, they have two claws, and they have a lot of characters that link them to some family in Dionycha, but every phylogeny, every attempted to put them in a phylogeny are really not conclusive. So the first one here is a morphology one, and these two in the middle, they are based on genetic markers, and this one is combined. and you can see how they vary the position of especially these three clades here in relation to the Oval Calamistrum Clade, the Maronites, and the other Dionychans. So we decided to investigate the phylogeny of Dionycha using comprehensive taxon sampling and also using a lot of data. We try to use genomic data and also morphology, transcriptome, and also the usual suspect markers. So that allowed us to have like a really comprehensive sampling of this group. And this like result, we did a lot of tests and a lot of inference methods, and actually this like they're very congruent depending of the matrix we use. And we did topology tests to test alternative hypotheses about the position of some clades, for example, Sparasside. And this is the position that we found as sister to Maronites. And we did constraining hypothesis tests, and they are significantly worse than this one. And so we ask ourselves, is this result caused by inference error? Like we know that sometimes when we have a lot of gene tree discordance, so the gene tree discordance could be because of incomplete lineage sorting, or just because you don't have enough variation in your genetics, genetic markers in your UCEs or whatever to infer a correct gene tree. So to test that, what we did is use a simulation. So we have this alternative hypothesis where Sparassidae is related to the other Dionychas. And then we simulate gene trees using coalescence process inside this containing species tree. And then we simulate sequence for each genes, and then we concatenate and then generate a species tree of this simulated data and repeat this 1,000 times. So, so just to remember what we want to answer. So the question we want to answer with this is, what are the chances to recover Sparasside assisted to Maronites in a model which Sparasside actually belonged to Dionycha, but evolved in a rapid radiation? And based on our results, the answer is 0%. So we never found in our simulation the Sparasside with as assisted to Maronites. That indicates that our results, it's not likely to be caused by inference error. We did the same for Thomisidae. So Thomisidae is always like inside the Oval Calamistrum Clade, and it's unlikely that this is an error of inference methods. And also the same for Mimicicultori. This is like a recent described family. But here we got a slightly different result. So we got like 80% of the simulations, they're found to be in this position, even though when there's the model simulator include them as Dionychus. So what we can conclude that, yes, this could be an error. So we are not so sure about the position of this family, but for now, this is the best position we have, but would be nice to have more data on that. And what about protodomini? Like, what the hell are the protodomids? And so every morphology so far, every work using morphology so far puts the protodomini inside Gnaphoside or really close related to Gnaphoside, including the most recent one published by Bruno and Chris Himes. The morphology tells that they are Gnaphoside, but it's not what molecules tell. And these are, in our analysis also, we show that protodomini is sister to the remaining Dionychas. And we did the same test, and this topology, like the topology test and simulations, show that morphological hypothesis is very, very unlikely. So this is the picture, like the big picture of Dionycha phylogeny that we have. The Dionycha is sister to the others, to the remaining Dionychans. The protodomini is sister to the remaining Dionychans. And we have two groups, the Dionycha B and Dionycha A. Dionycha B with salticids, corinids, and Dionycha A with the anaphanids, clubionids, and gnaphozoids in general. So with that, with this phylogeny, you have a framework to understand the evolution of some characters. So we can ask, how did the third claw evolve? And we did like several tests also using Bayesian ancestor state reconstruction and Bayesian testing of different hypotheses and models. And we found that it evolved at least 6 times, but one of the losses of the third claw wasn't the ancestor of Dionycha. So that could be a synapomorphy of Dionycha. And also we did the same for the claw-tufts. And also we show that it evolved like several times in the outgroup. So there's some convergence, but is a synapomorph of Dionycha also, with some losses here inside Dionycha A and Dionycha B. The same for the eyes. So we can see they evolve convergently in Mimesicultoria, here in Predominant, but it's like a synapomorphy here of Gnaphosidae, or could call like Gnaphosidae, but also evolve here once inside one trochanteriid. The same for the Piriform Gland Spigot. So we also show like this, it's the base of Piriform Spigot in Predominant. It's really, you can see here is like really a long base with a short shaft, and this convergently evolve here in Podracids and Molicrini with a really long base and then a short shaft. So it evolved at least 3 times. But like we also ask if those convergences are really convergence, or like are they really homoplasies, or are they hemiplasies? So what is an hemiplasy? It's a deep homology that it shows as it were an homoplasy because of a deep deep ancestral polymorphism that was incompletely sorted to the population. So it's a incomplete linear sorting of the genes related to the expression of those characters. So we test that using qualitative simulations also, and we found like for all the characters, only the base of the spigot could be explained by homoplasy. So here, like, so we would have one convergence here, but here we should have had a rise only once in hemiplasy way, so in the ancestral of all this group here, but then was incomplete sort through the other groups, other families inside this group here. So we also test the evolution of the clasping mechanism and the base. So we have this base that are movable. So some species lost this base. So the tooth, like the seta, are attached and not movable. So it could be harder for the spiders to remove it. And other species, as an alternative, they developed this clasping mechanisms here that would help to grasp the seta. So we were thinking if this would be like a way to overcome the problem of losing the base. So we tested with models, with comparing like different models of evolution. One independent model and one which those traits are correlated. So we found that yes, like it's most likely to gain this clasper or some kind of clasper when you lose the base here. So why do these convergent evolution events and then anaplasy? Why do you see these convergent evolution events? Most likely because of adaptive radiation at the end of Mesozoic caused by an ecological opportunity. But this is just a topic for other talk. And then you can see a little bit here in this link on YouTube that I talk a little bit about this work that we are doing too about the Dionycha evolution and adaptive radiation. So just like to finalize, we provide this robust phylogeny of Dionycha. The phylogeny serves as a framework to understand the character evolution of the group. And we show like some convergence and hemiplasy in the Piriform spigot. That's all. Thank you for everybody, and thank you all the collaborators and institutions.